Endomembrane trafficking is the flow of products between the organelles of the cells. I'll start off by explaining the organelles. The nucleus is the brain of the cell, and it contains the instructions for the cell, like DNA. The endoplasmic reticulum organelle is used for production and packaging. There are two types, rough and smooth. The rough contains the ribosomes, which produce the protein for the cell. The smooth ER produces the lipids. Ribosomes float freely throughout the cell, except for on the rough endoplasmic reticulum, and is where protein synthesis occurs. The Golgi apparatus is like the UPS of the cell, and it packages the final product of the proteins, either secretes the protein or sends it out of the cell. Lysosomes contain enzymes that break down waste in the cell. The plasma membrane is the outer layer, which is made up of the lipids and is amphipathic, which means both water-fearing and water-loving, and it also provides protection. DNA stands for deoxyribonucleic acid and is a double helix and is composed of nucleic acids adenine, thymine, guanine, and cytosine. Adenine and thymine pair up together, and guanine and cytosine pair up together. DNA replication is the first process of the endomembrane trafficking, and the double helix is ripped apart by the polymerase, and it makes a template strand containing the codons. A codon is 3 nucleic acids, which code for specific amino acids to match the nucleic acids. The template strand will be used for transcription, which is in the second process. Transcription is the second process where the DNA is copied onto a strand called mRNA. The mRNA will later be translated. Transcription is located in the nucleus. Thymine is no longer in the mRNA strand and is replaced with uracil. When the transcription process is done, the mRNA then travels to the rough ER where the translation will occur. Translation is where the RNA is read by the ribosomes and the amino acids attached to the tRNA. tRNA stands for transfer RNA, and it has an amino acid and anticodon attached to each end. Anticodon is a sequence of pairs that attaches to its other pairs. The ribosome holds the RNA in place while the tRNA attaches itself to the anticodons. Second tRNA attaches, and then a polypeptide is formed between amino acids and the tRNA. This process continues until the mRNA reaches its stopping codon. Ribosomes release the RNA when the polypeptide is formed to its fullest. Polypeptides go through the post-transitional modification before leaving the rough ER. Post-transitional modification of the polypeptides folds or bends the polypeptide before exiting the rough ER, and then from there they form into true proteins. Proteins are shipped in vesicles to the Golgi apparatus. Vesicles are like the backpacks of the cell, and they transport things around the cell. The vesicles enter the cis face of the Golgi apparatus and then are formed into the new vesicles, which are shipped out of the trans face of the Golgi apparatus. The Golgi is made up of flat vesicles called cisternae. They're like pancakes. When exiting the cell, it's going to a new process called exocytosis. Exocytosis is where the products from inside the cell are moved out of the cell, where the vesicles fuse to the cell membrane and then is secreted into the extracellular fluid. And that concludes endomembrane trafficking.